Right. Okay. So this one is just a picture, okay, to show how this particular toxic, okay, can increase in the aquatic system. Okay. So we have here, okay, we have the aviation industry, we have the urban and this agricultural source, okay, that will from the anthropogenic sources, okay, which is involving human activities. So we can start to see urban, industrial, and agricultural runoff will start to deposit into the aquatic and also the marine sediment. And this particular aquatic and marine sediment comprise of water. It comprises of sediment. Water is on the top part. Water sediment is at the bottom part. And by the biota is organisms that living in this water system, which include fish. Okay, we can see fishes here swimming here and there. And then crustaceans. Okay, crabs and prawns. You can see the prawns here and the crabs here. Mollusks and oysters, okay, around this area you can see the mollusks, okay. And also the deep part here. So what happened is this particular urban industrial and agricultural runoff will start to sediment and then deposited into this area, and it will be taken up by the organisms such as the prawn, the crabs, okay, and then the crabs and prawn will be eaten by the fish, and as well as the mollusk also will be eaten by the prawn and also the crab. So uh, all this particular runoff will be deposited into the tissues of the animals. And when we detect or when we consume these fishes, for instance, or the crabs or prawns or the mollusks, okay, we will also accumulate all the toxins within our tissues. And eventually, if the uh, concentration, okay, of these toxins increase in the body of the human who consume all these particular biotas in this aquatic and also marine area here. So it will start to cause disease, okay, towards human, and also could lead to cancer, for instance. Right. And then the earthworm density, okay, and biomass are strongly influenced by contamination. So therefore, the earthworm is can be used as bioindicator to determine acute toxicity. Okay, fish also is a good indicator of water pollution. Okay, microinvertebrates are often used as bioindicators because they are very sensitive to pollution, excess nutrients, increased turbidity, chlorine, and so on and so forth. Frogs, okay, are also considered accurate indicators of environmental stress and the health of the biosphere as a whole. If you see this kind of frog here, see multiple limbs of the frog at the bottom part, okay, the hind limbs, okay, are becoming multiple here, so meaning something had happened to this particular area and eventually to the development of this kind of frog. Okay. And the types of bioindicators, we have the bioluminescence, okay, one bacteria as bioindicator, can also be used. Okay. They are used to test water for environmental toxins. If there are toxins present in water, the cellular metabolism of bacteria is inhibited or disrupted. This affect quality or amount of light emitted by bacteria. It is very quick method and just take about 30 minutes to complete, but could not identify the toxin. Okay, right. And the name of the microbes I can use is Vibrio fischeri. Okay, so when we talk about bioluminescence, what causes bioluminescence? So we have everything in biological system or happen because of the enzyme and so substrate reaction. So it is due to the substrate and enzyme complex that is named as luciferin. So luciferin is the substrate and luciferase is the enzyme, okay, within the cellular cytoplasm. So luciferin refers to any light emitting compound. Where the word comes from the root lucifer, meaning light bringing in Latin. Okay, and bacterial luciferase is an auto-inducible enzyme. Auto-inducible means that the above at certain concentration, the luminescence operons will start to be translated to or transcribed and translated to encodes luciferase. It is important to note that the luciferin and luciferase complexes differ among species. Not all species has the same lucifer and also luciferin complexes. And this one, if you talk about the microbes again that can be used as bioindicators, This one you can read more if you wish to. So location marine. Okay, the example of the bacterial species is Photobacterium phosphorium. Okay, this one to detect the phosphorus level. Photobacterium legionnaire, Altromonas hanidae, Vibrio lodgii, okay, Vibrio fischeri, Vibrio hawaiii, Vibrio splendidus. Okay, this is one. Sorry, one is a number. Vibrio orientalis, Vibrio vulnificus, VVLI, coastal area, and then we don't have anything here. Terrestrial Xenorhabdus luminescens, and Vibrio cholera beta albensis. Okay. This one, the coastal, why I leave it here. I want you to search for it, okay, and give the name. And if the exam asks you to give the name, then you please do so, okay? Thank you. And then microbiome indicators and chemical pollutants. Microorganisms can be used as indicators of aquatic or terrestrial ecosystem health. And these particular microorganisms found in large quantities, microorganisms are easier to sample than other organisms. That's why we prefer to use microbes, because the size is small. It can be found in larger quantities, although the microbes are in large quantities, but you can just store it in a petri dish, for instance, because in a petri dish contain about maybe billion of organisms in there, cells, but because due to the small size, you can easily carry, and that's why microorganisms are preferred choice rather than the plant, rather than the animals. Okay, the algae is still good because you can get numbers, okay, billions of numbers, because the size is also in a micro size. And then some microorganisms will produce new proteins called stress protein when exposed to contaminants like cadmium and benzene. Okay, so this stress protein can be used as an early warning system to detect high levels of pollution. Okay. So these are all the proteins involved. Okay, you can just see. And then this one is okay example of the bioluminescence from by various species. We can see this one is from jellyfishes. Okay, and this one from the what do you call it? Click, okay? Or firefly. Okay, this one's from firefly. You can see up here is from firefly, and also we also have algae and the marine dinoflagellates. We call as blue tears. Siapa yang pernah pergi kat kampung Kuantan di Kuala Selangor, okay, boleh tengok blue tears. You disturb the water, the color become blue, and that one is due to the action of the luciferase enzymes from organism that is called as marine dinoflagellate. Okay, so summary. Okay, you can see as a summary for this chapter, you can say that plant indicators. The users is the presence and absence of certain plant or other vegetative life in an ecosystem can provide important clues about the health of the environment. And you can see that we have like that. Now, in animal indicators, you can observe the increase or decrease in the population. So we can see like the fishes, okay, the frogs, as I mentioned. Okay, you can read them yourself. And microbiome indicators, such as the one that I mentioned to you earlier, okay, previously in the slide. Okay, users of bioindicator. So how can we use bioindicator? The first one we need to do is we want to detect the changes in the natural environment. Number two, we want to monitor the presence of pollution, okay, and its effect to the ecosystem in which the organism live. If the organisms start to die, then yeah, we can say that the pollution is very dangerous and it could harm the human as well. And then monitor the progress of environmental cleanup and test substances like drinking water and for the presence of contaminants. Okay, this is also important, okay, for us to determine. And then okay, and then the most important reason, okay, for using bioindicators are the direct determination of biological effects. Okay, we want to know the biological effects. Okay, whether there is or there are not. Okay, there is yes or there is or there is no. And then the determination of synergetic and antagonistic effects of multiple pollutants on an organism. So sometimes this compound